capable, committed people who are experts in their fields. That's directly linked to our success.
We are one of a handful of universities in Africa that also provides training up to a PhD level, reflecting that we are future-focused.
Our advanced infrastructure and technology enables us to actually compete with the best in the world.
The lab story started in the mid-1990s with Professor Peter Smith. He was approached by the South African Medical Research Council to conduct a bioequivalence study on the first-line TB drugs. And as we know, Professor Smith, very positive, optimistic person, he took it on. He assembled a team of laboratory scientists and clinical scientists, and they successfully conducted the first bioequivalence study at the Division of Clinical Pharmacology. And after that, many of these studies followed. Today we work with pharmaceutical companies, CROs, NGOs, clinical trial networks and global health partners around the world.
So over the years, our analytical capabilities have actually expanded quite significantly. We moved from using HPLC UV analysis to highly sensitive LC-MS technology with about 10 workstations that we actually have. They allow us to support increasingly complex clinical research studies and also analytical challenges. Our work has grown from first-line tuberculosis medicines to antiretrovirals, antimalarials, second-line TB drugs, and a broad range of other therapeutic compounds. We develop and validate methods not only in plasma, but also in other challenging matrices. Working with complex drugs and difficult clinical samples have actually taught us to be quite adaptable. Each molecule behaves differently, and each matrix actually presents its unique set of analytical challenges. So our experience actually allows us to design methods that are not only scientifically robust, but also practical in the realities of clinical research.
As our science developed, a dedication to quality developed with it. Our laboratory participates in international proficiency testing and quality assurance programs, and our research has contributed to treatment optimization, policy development, and international guidance, including WHO treatment guidelines. Our laboratory is ISO accredited and operates in accordance with GCLP and GLP principles. Our computerized systems are 21 CFR Part 11 compliant, and our assays are validated in line with current international bioanalytical standards, including FDA and ICH M10 guidelines. These quality milestones reflect an important principle. Reliable bioanalysis depends on more than just advanced instrumentation. It requires trained people, quality-controlled processes, scientifically justified methods, and a culture in which data integrity is never compromised and quality is the norm. For our clients, dependable bioanalysis begins well before the first sample is analysed. We work with study teams from the planning stage to align the analytical strategy with the study objectives and provide guidance on sample collection, stability requirements, logistics, timelines, and reporting needs. Early engagement with the client really helps reduce analytical and operational risk, particularly for complex studies. Once samples arrive at the laboratory, they move through controlled processes for receipt, storage, analysis, data review, and reporting. At every stage, our focus is on traceability, data integrity, regulatory confidence, and clear communication with the clients. Our greatest strength is our team. Through our honours, masters, PhD, and postdoctoral training programs, we are also committed to developing the next generation of bioanalytical scientists, strengthening both our own laboratory and the broader clinical research and drug development environment. Our operational model is scalable, enabling us to assay 40,000 samples annually. We have the capacity to support large demanding studies while remaining specialized enough for the client to engage directly with the scientists responsible for their work.
Well, it started with bioequivalent studies to help develop the generic drugs. And today the focus is more onto the clinical trials on phase 1, 2 and 3 studies. We're getting into biomarker discovery and development work. And we also introduce methodologies to analyze bigger molecules like peptides and proteins, antibodies, biosimilars, those kind of molecules. We aim to provide confidence. Confidence in the method, confidence in the data, and confidence that the bioanalytical component of the study is in the hands of experienced professionals.